Hello everyone, and thank you for taking the time to view my presentation at the 2021 Undergraduate Research Symposium. In this presentation, I will discuss how this summer, I, along with the help of my faculty mentors, Dr. Susan Pagano and Dr. Kara Mackey, used mathematical modeling to assess physiological condition of thrushes. I'd like to start by introducing you to the main characters in today's story, Swainson's, gray-cheeked, and hermit thrush. These three species are all of the genus Catharus. These thrushes breed in northern areas of North America and migrate across the U.S. before reaching non-breeding territories in the southern U.S. or Central and South America. In this project, our focus is not on the locations these birds migrate to or from, but rather on the places these birds stop to rest and refuel at during migration, called stopover sites. Of special interest is a stopover site that is located about 20 miles north of RIT on the south shore of Lake Ontario. Located here is Braddock Bay Bird Observatory, a bird banding and migration monitoring station. As you can see from these capture date ranges, thrushes arrive at Braddock Bay during fall from a rather variable window of about late August to early November. This diagram provides an overview of the bird capture and blood sampling process at Braddock Bay. From left to right, you can see a mist net used for capturing birds, the measurement and banding process, the separation of blood plasma from red blood cells using a centrifuge, and the measurement of plasma metabolite concentrations using assays in a microplate spectrophotometer. Using the Braddock Bay dataset, we developed the project with two pieces. For our first piece, we built models to estimate fat levels in Catharus thrushes. For our second piece, we took the opportunity to analyze a smaller subset of plasma metabolite data. So you may be wondering, why are fat levels and plasma metabolites worth studying? Well, fat mass is an important measure to ornithologists, as the primary energy source for migratory birds is fat. However, measuring actual fat stores in birds often requires sacrificing birds or more complicated methods. We wondered if we could use mathematical models to estimate fat content. As for the metabolites, it has been found that certain plasma metabolites, such as triglycerides, are essentially stand-ins for instantaneous rates of mass gain in birds. Kind of like a derivative, if your brain is more tuned with calculus than biology. Scientists are interested in these rates as they signify how quickly birds refuel and thus reveal important information about stopover site quality. We wondered if observing blood plasma data and fat levels could help us determine if there are species differences in how Braddock Bay is used as a stopover site. Additionally, we hypothesized that statistical analyses of blood plasma data could potentially reveal year-to-year variation in metabolite levels and stopover site quality. Now that we've discussed some background information, I'll jump into some of our methods. I'll start by discussing the methods we use to predict fat levels in birds in the Braddock Bay dataset, and then I'll talk a little bit about how we analyze plasma metabolite levels in a smaller subset of these birds. And for reference, all analyses in this project were conducted in JUMP Pro 15. We started by dividing birds into classes based on wing length, a common measure of body size in small birds. And this was done separately by species. We made note of all wing length classes that had 12 or more birds in them. Once we divided birds into wing classes, a regression model using the numerical fat score that bird banders assign a bird to quantify its fat reserves was created for each wing length class. Fat score was treated as a predictor of body mass. Once the regression model was made, our next goal was to derive estimates of fat-free mass for a specific wing length. As seen in this right-hand figure, we did this by selecting all data points that fell within a 95% confidence interval of the intercept of our linear regression model. After we built regression models for all wing classes and selected data points representing our estimates of fat-free mass, we built a second type of regression model with wing length as a predictor of fat-free mass. We then derived an equation to represent the regression line. The equation we derived was then used to calculate fat-free mass for all birds in the dataset. Fat-free mass was subtracted from overall body mass, then 2 grams was added to this number. This was done because per prior studies, fat scores of 0 often fail to account for around 2 grams of fat in birds. So we added 2 grams to help account for this potential error. And we had done it. We formed an estimate of fat mass for each bird in our dataset. The last thing we did with our fat mass estimates was compare them to condition indices, often used to represent fat stores in birds, by looking at correlation coefficients. And I'll discuss the results from this a little bit later. Moving on to metabolites. Before this project even began, metabolite concentrations of triglyceride, glucose, and uric acid were recorded for a subset of birds in the dataset we used this summer. We took two different approaches to our metabolite analysis. We performed multiple regression with backward selection to see if things such as capture hour, bleed time, year, week of capture, or scale mass were significantly related to our metabolites. We then used ANCOVA, including significant independent variables as covariates, to assess yearly variation in metabolite levels. For the second track of our metabolite analyses, we ran a principal component analysis of triglyceride, uric acid, and glucose. We interpreted our first principal component score as a refueling index of birds at the stopover site. We then used ANOVA to see if there was significant variation in this refueling index across years. I will now move on to my favorite part of the scientific process, sharing the results. We started this project by looking at some summary statistics for both the Braddock Bay dataset and the smaller subset of blood sampled birds. We noticed right away that our summary statistics for the small dataset were quite similar to statistics for the large dataset. This helped convince us that our small subset of birds was likely representative of birds in our larger sample. We observed some interesting species differences in our summary tables. For example, Swainson's thrush and gray-cheeked thrush have a median day of capture in late September. However, we found that hermit thrush arrive at Braddock Bay later in the season with a median capture date of October 15th. These differences in migration timing could potentially affect how the different species are able to use Braddock Bay as a stopover site. This bar graph provides an overview of both the mean grams of fat mass we estimated for each species, as well as the mean fat mass percentage. Similar to the size ordering of species observed in our summary statistics, our estimates showed that hermit thrush had the fewest grams of fat, while gray-cheeked had the most. However, fat mass percentage was higher for Swainson's compared to hermit and gray-cheeked. This could potentially indicate that Swainson's have had more success in building and maintaining fat stores than hermit and gray-cheeked thrush at this site. This table on the right shows the correlation coefficients of fat mass estimates with 5 condition indices used to represent body fat content in birds. We observed statistically significant correlation of fat mass estimates with each of these indices. Something interesting we found is that some of the condition indices were more highly correlated with our fat mass estimates than others. The residuals of an ordinary least squares linear regression model of wing length and body mass, for example, show nearly perfect correlation with fat mass estimates. Future work to verify our estimates is necessary. If in future studies we find evidence that our models are accurate at predicting fat stores, then we could potentially use our fat mass estimates to help us determine which condition indices are the best predictors of fat content in thrushes. Now that we have looked at some results from the fat mass estimation piece of this project, I'll talk a little bit about metabolite results. This table displays independent variables that were retained as significant in our multiple regression models with backward selection. Without getting into too much detail, our most important takeaway from this table is that year was retained as a significant independent variable for all 3 metabolites and for all 3 species. This supported our idea at the beginning of this project that year-by-year variation in metabolite levels could exist. This graph shows how our refueling index of triglyceride, glucose, and uric acid combined varied by year. What we feel is important to note from this is that there seem to be differences in each species' ability to refuel in different years. However, all 3 species were shown to follow a similar yearly trend in metabolite levels. An ANOVA test revealed statistically significant yearly variation. Additionally, our post-hoc Tukey's test showed significant differences between different pairs of years. Most notably, 2013 was shown to be significantly different from each other year, and this pattern was observed for each of the 3 species. So, our results suggest that there was something happening in 2013 that was affecting refueling abilities at Braddock Bay. Overall, our idea that yearly metabolite variation exists was supported. So, how did our results help us answer our questions at the beginning of this project? What can we conclude? As you'll remember from this presentation's introduction, Our project really had two main research questions. First, we were interested to see if we could determine if there are species differences in how Braddock Bay is used as a stopover site. Well, variation in mean fat content and fat percentages, as well as differences in metabolite levels, do seem to indicate that species differences exist. And second, we wondered if we could observe yearly variation in refueling as measured by plasma metabolite levels, and if so, is there any way that we can explain this variation? Well, our backward selection results, as well as similar trends in metabolite levels for each species, do support the idea that yearly variation exists. However, we can't conclusively say at this point what could be causing this variation. But we do have some ideas. This is a graph showing the mean temperature for the month of September from 2013 to 2019. September is the month for which over 50% of the birds in our dataset were captured. We noticed that this yearly variation in average temperature is quite similar to the trend we observed in metabolite levels. For example, 2013 has a lower temperature for September than the other 6 years, and 2013 was also the year for which all of our species had significantly lower refueling indices. Future work is needed to help us understand how environmental effects such as lower or higher temperatures may affect migratory birds. However, similar metabolite level trends and average temperature trends are encouraging for those of us trying to understand what factors may be influencing migratory bird health and stopover site ecology. On a broader scale, and this affects you, even if you're not directly interested in migratory birds, looking at climate trends compared to migratory bird condition trends could even help us learn about some of the ecological impacts of climate change.